The same logic used to generate software and text is now being applied to the physical assembly of DNA. We are seeing artificial intelligence produce the precise instructions required to build living cells. In a recent laboratory experiment, researchers used a generative model to design complete, functional viral genomes that do not exist in nature. When these AI-designed sequences were synthesized, they functioned exactly like their natural counterparts. These particular creations are not human pathogens. The experiment used bacteriophages, viruses that only infect bacteria, to demonstrate that machines can now author viable biological code. This milestone suggests that biological systems, once viewed as uniquely complex, are increasingly becoming programmable through machine learning. This work was led by researchers at Stanford University and the Arc Institute. They utilized EVO1 and EVO2, foundation models trained on more than 2 million genomic sequences, rather than human prose. The team focused on the bacteriophage, a class of virus that hunts and kills specific bacteria. These viruses have been studied for a century and are safe for laboratory use because they cannot infect human cells. Because of this host specificity, bacteriophages serve as a controlled proxy. They allow scientists to test whether an AI can write a working genome without the risks associated with human infecting viruses. Refining these harmless bacteria eaters provides a template for engineering microscopic systems with a high degree of precision. The researchers prompted the AI with a tiny snippet of phage DNA. The model generated 700,000 potential genome designs, each a unique variation. Computational filters narrowed this pool to 285 designs likely to function. These were then physically synthesized into DNA molecules in a lab. When tested against live cultures, 16 of these machine-designed genomes successfully infected the target bacteria and began reproducing. One design contained 392 distinct mutations and successfully integrated proteins in a configuration that human engineers had previously failed to achieve using traditional methods. The model arrived at these complex biological solutions by bypassing the slow pathways of natural selection, producing functional architectures in a single day of computation. This capability addresses a major challenge in medicine, the rise of superbugs like E. coli that have evolved resistance to nearly every existing antibiotic. In the Stanford experiment, a cocktail of AI-designed phages was deployed against these resistant strains. The synthetic viruses latched onto the bacteria and annihilated them. Traditionally, developing a new treatment requires mining nature for rare, effective viruses. This experiment demonstrates a shift toward prompting biology for customized, instant solutions. While limited to a laboratory setting, the success of these synthetic phages suggests a method for responding to bacterial mutations with machine-designed solutions at a much faster pace. This breakthrough introduces a significant dual-use dilemma. The same generative pipeline used to target lethal bacteria could be repurposed to design viruses that target human cells. Current biological defenses rely on homology-based screening. When a lab orders custom DNA, software checks the sequence against a database of known threats to see if the order looks like a recognizable pathogen. However, AI can generate novel sequences that function like bioweapons but share no structural similarity with known viruses. These sequences can pass through existing scanners entirely undetected. Regulatory oversight has not kept pace. While Congress debates legislative fixes, benchtop DNA synthesizers continue to be sold without mandatory software to check for these novel, functional threats. Biological defense infrastructure is designed to combat known threats. It is structurally blind to the synthetic, machine-authored pathogens of the future. There are currently practical limits to this technology. The majority of the designs produced by the AI failed to function, and current models are not yet capable of rewriting complex human pathogens without breaking them. A digital blueprint also requires physical synthesis. AI cannot manifest a virus into the real world without access to specialized equipment and human intervention. Those barriers will likely lower as the technology matures. This leaves a window for lawmakers to mandate function-based screening, systems that analyze what a DNA sequence does rather than just what it matches. As the boundary between software and physical biology dissolves, the ultimate question is no longer what AI is capable of creating. It is whether our governance can adapt to manage it.